standing in between you guys in a break, so let me know if I'm not talking loud enough, and I'll try and keep this going. I'm Grace Brown. I'm excited for this opportunity to share about some undergraduate research that I did in Dr. Piccolo's lab at BYU about metadata and machine learning. Public data is absolutely incredible. Funding agencies and journals require researchers to put their data on online databases so that other researchers can validate what they've done and reuse their data to address novel hypotheses. And there are hundreds of thousands of transcriptomic datasets in the public domain. However, these datasets are not of much use to other researchers without their metadata. Metadata is information about individual samples. Some examples of metadata include the tumor stage of a patient, the biological sex of a participant in the study, or the self-described ancestry of an individual. And scientists care about metadata because that information about samples may be their primary variable of interest. Many times, this metadata is collected so that you can control for confounding effects. And society cares about metadata because collecting good metadata enables precision medicine findings. Precision medicine seeks to not only take into account the symptoms someone has, but factors personal to their genetics and environment. So examples could be a patient's age and whether or not they have a wild type or mutant gene at a certain locus. However, there are two main problems with public metadata. The first is that there's a lack of standardization, so many terms are used to describe the same thing. All of these tables show real metadata that describes biological sex. As you can see over here, sometimes the terms alone contain the information that you need to determine biological sex. Sometimes you need a combination of metadata attributes and metadata terms, and sometimes the relevant metadata is mixed in with other metadata such as age. And this means that keyword searches alone won't pull all the data sets that have metadata a researcher is interested in. The second problem is one of sparsity. In our research, we found that approximately 13% of data sets recorded biological sex information. So most data sets are lacking that collected. So it's tricky to identify whether a data set contains the metadata a researcher is interested in. And we lack a way to do this reliably without going in by hand. So we decided to train machine learning models to determine whether or not a data set contains a metadata variable of interest. And we started by training a model to recognize self-described ancestry metadata, which is often represented by race or ethnicity terms. And while we started with self-described ancestry, we expanded our project to pick up on biomedical concepts that were easier to identify than ancestry and harder to identify than ancestry. The biomedical concepts we chose were biological sex, which contains fewer unique values, and tumor stage, which we thought would be harder to identify because there's many studies that are about, not about cancer, that record stage of disease information. So I'm going to dive into our experimental design. We'll walk through the different parts of this chart, starting with data collection. The data we used in this project was collected from BioProject and BioSample, which are run by NCBI, and they house metadata for public data sets like those in the Gene Expression Omnibus. Each project in BioProject represents a study, and information about each sample used in that study is stored on BioSample. The Gene Expression Omnibus is the largest transcriptomic repository, but recently studies from GEO are being stored on BioProject and BioSample. So these data sets are an organized subset of transcriptomic available that's publicly available. We downloaded all the transcriptomic Homo sapien data sets that were available on BioSample and BioProject, and at the time this was just over 27,000 data sets. We randomly selected 2,000 data sets to label by hand for our training set. Here are a few notes about our hand labeling process. Two of us researchers hand labeled the data sets independently of each other. Then we came back together to compare labels. And if we had disagreements, we went back to the BioProject to reach a consensus. If we still were unsure, we would go to the paper associated with that BioProject. And if we had further questions, we consulted Dr. Stephen Piccolo. Another note is that we tried to avoid making assumptions One example of this was with cell lines. The HeLa cell line is a pretty famous cell line from a woman named Henrietta Lacks. However, not all cell lines are as famous as hers. So if cell line was the only information that we had, we researchers did not make assumptions about the sample's sex, self-describing ancestry, or tumor stage. After labeling the training sets, we used cross-validation to compare two methods of machine learning inputs. The first method that we used was n-grams, which is pretty much subwords. We broke the metadata attributes and terms into subwords of length 3 and gave the model a binary profile of the presence of different subwords. One nice thing about this method is that it's interpretable to humans. So if you see that the subword THN is really relevant, we see that word or that subword in something like ethnicity, and we can do a mental check that things are the things that it's pulling out are meaningful. And then the other method we tested was embeddings. Embeddings are famous in large part due to ChatGPT. And we used the pre-trained Roberta model from the Hugging Face library to obtain embeddings. An example of embeddings in action is if you take the embedding for king, subtract boy and add girl, you get the embedding for queen. So there's some word relationships that are in these vectors and numbers. And precision and recall were mentioned earlier in this conference, but we evaluated model performance with these metrics. A common metric for machine learning is area under the ROC curve, but we decided to use area under the precision recall curve instead because it's better for data sets that have large class imbalance. Precision answers the question, what fraction of data sets the model says are relevant are actually relevant? And recall answers the question, how often is the model catching data sets that are relevant? So after finding the best form of input data via cross-validation, we trained a random forest model on the training data set, the 2000 labeled data sets, and then made predictions for the remaining 25,000 data sets. And we published those predictions back in January on Open Science Framework. After doing that, we wanted to evaluate precision and recall on those predictions, so we hand-labeled all metadata that was predicted to have a relevance of at least 10% to get precision, and then we hand-labeled 3,000 of those 25,000 data sets to obtain our recall statistic. And then this is the last part of our project. We took everything we labeled throughout the course of the project, the 2000 training sets and all the validation data sets, to quantify representation. It's been said that males are studied more often than females, and people of European descent are studied more often than those of other ancestries. So we decided to explore to what extent that's true in this data set. And this quantification just represents one potential application of our method. Let's go to our results. Here are the results from cross-validation, where we compared n-grams and embeddings as inputs. Surprisingly, we found that regardless of the biomedical concept of interest, n-grams performed better than embeddings on this task. And this may be because metadata is often short words or phrases rather than complete sentences, and embeddings are trained on sentence information. A few other considerations. We modified the class weight parameter of the random forest model to account for class imbalances, but found no change in performance. We experimented with n-gram profiles being a count of occurrence rather than just a binary presence, and found that that did not help cross-validation performance. We also experimented by putting in all of each bioproject's metadata as a whole to the model instead of each metadata attribute separately. While it takes longer to evaluate metadata at the attribute level because you're running more things through the model, this actually speeds up quantification on the other end because we know exactly which line is relevant to the biomedical concept of interest. So knowing this, we saved the n-gram trained model and ran the 25,000 not training data sets through degenerate predictions. And then using the validation data sets, here's how the model did. First, I'd like to point out that model performance depended a lot on the biomedical concept of interest. And like we expected, biological sex was easier to classify than ancestry, and tumor stage was harder. But overall, model performance for ancestry and sex were really high. Keep in mind that these numbers come from using less than 10% of the initially downloaded data sets for training. So if we increase that, we might even increase performance. Here are a few examples of where the model made mistakes. This was to identify ancestry. One thing to point out is that the n-grams that would come out of these metadata terms are not super common, so some of them may not have been present in the training set. And you'll notice that the concept of interest was mingled with other metadata information, which is just more challenging. So potentially by increasing the size of the training set, we can see a difference in performance. And now I'll show you the results from subgroup quantification. So this is all 27,000 data sets. What was the sample representation like? It appears that males are represented slightly more than females. Not too much to say here. And then when it comes to self-described ancestry, representation differences are much larger. Most transcriptomic samples came from people who described themselves as of European descent. And while the US runs bioproject and biosample, please keep in mind that this database has an international data source. Charts like this are helpful because if we want to be more inclusive, this shows us where we can improve. A few limitations of our method are that we only looked at transcriptomic data on this database. We only used embeddings from Roberta, but there's lots of other language models that we could have chosen to use embeddings from. We used a very simple machine learning model design, the random forest, as a proof of concept. We acknowledge that there's lots of other model designs that could work better for this task. And lastly, some data sets were really tricky to classify. So we want to be transparent, and we have our examples open to the public, so you can look at the decisions we made, and if you want to make them differently, we respect that. And here are a few applications of what we've done. The first one up here, we demonstrated for sex and ancestry. We can quantify subgroup representation a lot easier than if we had to do it by hand. In short, this allows us to see what's been studied a lot and what has not been studied too much yet. Machine learning helps us do this quickly. We can identify data sets that contain certain metadata variables, which is important for researchers that want to perform meta-analyses. And third, if you remember, one problem with the metadata was sparsity. Only 13% of the data sets included biological sex information. Ancestry and tumor stage were recorded even less often. And so maybe we can impute unreported metadata. If that subject interests you, you can stop by my colleague Tolu's poster. She's B40. And I'd like to thank the Travel Fellowship for allowing me to come here and present research. I'm also really grateful for BYU's College of Undergraduate Research Award funding this research. Thank you for your time. I'll take any questions.
Great, so we have time for a couple of questions for Grace, if there are any.
Go for it.
So you talked about word2vec, and you showed this very compelling example that we all love about, you can do algebra on it, and you can go from king to queen to, but then once you switch to other kinds of domain areas, it's what does that notion of analogy mean? And I wondered, when looking at your metadata, did you anecdotally or actually go and look to see what does it mean to do that kind of arithmetic on, say, tumor stage? Are there any analogies you can discover?
Yeah, that's a really great question. We did not do any sort of arithmetic stuff on the biological metadata that we had. That would be really interesting to do and see if there's a certain, yeah.
You can tell us next year.
Yeah.
Other questions?
I was just going to say, it seems like the inference and the embedding approaches would be complementary. We thought about like they might be discovering different pieces of relevant information. We thought about combining them at all.
Yeah, it's a really good question.
Can you repeat that question? Sorry.
Yeah, they asked if we'd considered doing a combination of n-gram and embedding inputs to the model. We haven't done that, but that would be a really interesting thing to look at.
Yeah, one last question. Yeah.
So just looking at the, so from your intuition, do you feel that you can design some guidelines how to design the metadata such that if the information there is actually less difficult for example, machine learning models to make a mistake, but if, even if, so let's say that there's no standard, can you at least improve
Yeah, I think that's a really exciting potential future application is that we could standardize what's out there to more accepted terms. I think that would be a really good application. We haven't done it for this, but it would be awesome. Yeah.
One quick question for Maria.
So I was wondering how how can you solve this? How well if you try to solve this task as like a zero shot LLM type of task? Here's my column header. Here's what is written and classified into the structured vocabulary. How well does it do? And can you use that framework?
Yeah, that's a great question. So, would you still want to encode it like with the LLM, like you said?
Well, I'm just like, you can take an existing LLM and just ask it to do this. How well does it do? Like on that very complicated example with the Russian mail, blah, blah, blah. How well would it
Yeah, that's a
classify
good
that
question.
into the structured vocabulary?
Yeah, it might work really well. Like if we just asked ChatGPT, does this have this sort of thing? It would probably get it.
I mean, do you feel like that's a good way to go forward with these kinds of tasks?
I think there's been a lot of advancements in LLMs, and I think they have a ton of applications to biology. And for things like this, I think they totally could help speed up a researcher's process. Yeah.
So let's thank